Well, good afternoon, everybody. If I'm not loud enough, please let me know, and I can scream. I'm going to give you just a short intro, and then mostly I'll be doing live demonstrations using a website that is freely accessible by anybody. So the idea is that if it doesn't work while I'm showing you, then that it doesn't work for you. So it should work. As Kristen mentioned, my name is Omar Harb. I direct scientific outreach and education for a database group called PathDB. PathDB stands for the Eukaryotic Pathogen Databases. And just to describe what this resource is, about 11, maybe actually 15 years ago, the National Institute of Health came up with a plan to put together resources around the country that are dedicated to bioinformatics. And they developed, ultimately ended up being 4 resources around the country. One of them was for bacterial organisms. One of them was for, so one of them was for viruses, so things like the flu, Zika, for example. Those would fall under the Viper resource, or viral pathogen resource. There's one for bacteria. The database is called Patrick. All of these are freely accessible. They're all funded by the government, by my taxes, your taxes, fund these resources. And then there's one for vectors of all of these organisms. So bacteria, many bacteria need to be carried by a vector, like a mosquito or a tick. Same thing with viruses. And ultimately there's PathDB, which is a database I work for, which is for things known as eukaryotic pathogens. So we're all familiar with the difference between a eukaryote and a prokaryote, and a virus, obviously. And eukaryotic pathogens are basically eukaryotes that are actually pathogenic. So things that can cause disease in humans and animals. And there are many of them, unfortunately. And these include things like Plasmodium, which causes malaria. And so that causes almost a couple of million deaths annually, and about half of the world's population is living in endemic regions. There are diseases like Leishmania, which many of our troops who've gone to Iraq, they come back and they have cutaneous leishmaniasis, which is caused by this parasite called Leishmania. There's sleeping sickness, you've probably heard of that, which is caused by trypanosomes. That is not a terrible disease. It's a terrible disease in humans when they get it, but it's actually more of a serious problem because it affects cattle. And in many parts of Africa where it actually is endemic, it actually is a livelihood. So a farmer would have his herd of cattle, or maybe one cow, and that's their livelihood. And imagine that your livelihood is, dies because of a parasite, right? That destroys not only the cow, but also your potential to survive and be productive in your society. So many of these diseases actually not only cause death and morbidity in humans, but also to the animals and the things we depend on to survive and live. PathDB not only includes sort of your traditional eukaryotic pathogens like Plasmodium, it also includes things like fungi. And fungi, we know fungi are ubiquitous, they're all over the place. And so there are resources for fungi as well within this umbrella of databases. The philosophy of all of these databases, and in particular PathDB, is really to try and take all of this data that's being generated by scientists all over the world and put it into a resource, into a database that makes it very accessible by the basic scientist, the scientist who wants to ask questions about that underlying data. So imagine if you want to go shopping, right? And every time you want to go shopping, you have to go down to the Italian market in Center City or in downtown Philadelphia, and you have to go from one store to the next, and you have to sort through all the products that are in there. And then at the end of the day, you say, okay, I went to 5 stores. Out of the 5 stores, I found, this store had t-shirts, that one had, pants, this one had t-shirts of this color, this one had t-shirts with pockets. And now I'm going to go back and I want to go put together my suit because, I'm going to buy the t-shirt that was the cheapest here, I'm going to buy the tie that was the cheapest or made out of the best material from this store and so forth, right? It'll take you all day long, and you manually have to go in and do this sort of accessing the database of the stores, right, that are there. But, fortunately or unfortunately, depending on your viewpoint, you can go online and go to Amazon, right? And you can quickly go in and filter your products, add them to your basket, and again, within maybe the span of 15 minutes, you already have a complete suit put together from probably a shirt that's made in China and delivered from China, pants maybe that are made in Tennessee in a plant there, and maybe shoes that are made by a company in Seattle, right? So, and then you get them all delivered to you, right? So that's the beauty of having a database that allows you to access all of this information. And these databases, the scientific databases, are essentially the same thing. The goal is really for scientists to provide you with the ability to run these searches in a database without having to manually go to the stores, manually access the files on from various resources and analyze them yourself and put the data together. So the databases, these types of databases are trying to make it easier for you to access the underlying data. And really, ultimately, it's to enable researchers to ask their own questions. So databases like the resources like the ones that are in PathDB, we include data from many scientists around the world. So if there's scientists out there working hard, producing all of this data, they publish a paper and they show you these beautiful figures, right, with a graph going up this way, images of a parasite doing something or a cell doing something else, and they show you the conclusions, right? But what's feeding all of these amazing images, these amazing graphs, is millions of rows and columns in an Excel file, for example, right? And that's a wealth of information that's there that's not being presented in the figure. The figure is just a summary of everything that was in their conclusions of the data. But the data has a lot more in there that you can mine and you can ask questions about. And so these resources allow you to go in and start asking your own questions about the data. Of course, you have to have some fundamental basics about the biology in there. And we're all here where we know biology, so I think we're all pretty much equipped to start asking some basic questions using resources like these. And hopefully you don't get intimidated just that, you know, there are scientists out there doing hardcore research in labs and they're using these resources. It doesn't mean that anybody can actually use these resources and know something about biology, and start asking some basic questions and really coming up with something that's testable in the lab. And in fact, many of the users of our databases actually do that. They run a series of questions online, and then they test the question they asked online in the lab and see if it works. So imagine if you've on Amazon again, you put together your suit, and you think, I think this is going to look good. Ultimately, your test is you order the stuff, and you put it on, and you look in the mirror, and you say, oh yeah, this looks good, or it doesn't look good, right? And you go back to the drawing board. And that's the same thing here. Scientists ask these questions, and they go to a lab and run the experiment, and they get results that prove or support or don't support their hypothesis, and then they can go back and redo their in silico work. Okay, so I'm going to switch to a live demo of one of these websites, and we'll then move towards the modules that Kristen talked about, which are based on a database called ToxoDB. And so again, you can access any of these resources from any web browser. And in this case, we're going to use ToxoDB, which is toxodb.org. That's the website, very simple. Okay, we're going to be using a small part of ToxoDB, or one part of ToxoDB, which is the Genome browser. And the idea behind this is that using the Genome browser, you'll be able to illustrate to yourself and to your students how one can look at a genome, right? And look at genes on a genome, and then maybe try and tackle and understand some data that you can paint on the genome, and sort of that allows you to, and gives you some evidence about the genes and the different information about a genome, the life cycle of a parasite, and so forth. So before I go too much into the database, so how many people here, how many people have looked at the module online so far? Just out of curiosity. Okay.
Right.
All right. So how many people have heard of toxoplasma before? So toxoplasma, just so we're all on the same page, it's a parasite, again, a eukaryotic parasite that has a nucleus, just like our cells. And it is ubiquitous. It's pretty much everywhere. So a third of the people in this room are infected, right? I'm probably infected. It depends, you know, I love to eat meat, and sometimes on the raw side, definitely infected, right? And that's, you know, I'm guilty. But, and depending on where you grew up, which part of the world, if you visited parts of the world where you've eaten meat or vegetables, if you're a vegetarian, don't worry, you're probably infected as well, because there are other routes of infection too. So So Toxoplasma is a really interesting parasite. One is that it infects such a large number of people. Some parts of the world, like in France, for example, or Indonesia, for example, it's up to 80% of the population is infected. You're a carrier of that parasite. It is living in you, okay? Now, it sounds really scary. Oh my God, I'm carrying a parasite. Well, it turns out we carry lots of parasites and viruses and bacteria all the time. And it's just that our bodies have really co-adapted, co-evolved with these parasites and organisms, and we sort of are living in this, let's say, harmony, but you're controlling that infection. So the parasite is living in you, but your immune system is able to control it and prevent it from going haywire. Of course, when your immune system goes kaput, obviously, right, that's when things could go wrong. And it turns out that Toxoplasma was a leading cause of death in AIDS patients back when the epidemic was at its peak. Because as soon as your immune system shut down, these parasites would reactivate and start growing. And in fact, Toxoplasma in mammals, it actually forms these, what I call cysts, in various tissue. It could be in your muscle tissue or in your brain. But it's completely under control by your immune system. But when your immune system stops working, then these cysts will reactivate and start growing again. And it's that rapidly growing parasites that actually cause the disease, right? So if you have a cyst in your brain and the parasite starts growing like crazy in there, it just eats up all your cells, right? And in fact, when you look at scans of AIDS patients who died from Toxoplasma, there's essentially a big hole in their brain where Toxoplasma was growing and replicating. So it could be a very serious disease in humans. It's also a very serious disease for the fetus, the unborn fetus. So that's why you probably heard people telling women who are pregnant to stay away from cats, right? Don't get scratched by a cat. Don't change the kid litter, right, when you're pregnant. And that is because cats are carriers of this parasite. And not only are they carriers, they actually do a very important thing in the life cycle of the parasite. So all of these parasites, actually any organism on life has a life cycle, right? And depending on how you paint it, it can sound very, very complicated or very, very simple, right? Humans have a life cycle, right? Where our fetus develops in a certain way, and we're born and we grow, and then we die. There are various developmental stages in our life cycle. Same thing with a parasite, it has developmental stages, right? And these parasites do something a little bit different. So when they grow in our bodies, or in a sheep, or in a cow, or in a rodent, right? They just replicate themselves, and then they get stuck in a cyst because your immune system takes over and stops them in a cyst. And that's pretty much a dead end for the parasite, unless you're eaten by another animal, right? And then the cyst can be transferred. Cats, though, are really interesting when it comes to Toxoplasma. So they can get infected with Toxoplasma, but what happens, Toxoplasma undergoes sexual reproduction inside of the cats. Okay, that's where genetic exchange can happen between different Toxoplasma parasites. And that's where it forms something called an oocyst. An oocyst is a very environmentally resistant shell that carries parasites inside of it. And so ultimately the cat will shed the oocysts in its feces, and that's why they tell women not to mess with the kitty litter, because that's the most likely location of these oocysts, which are highly infectious.
Why cats?
Nobody knows. Very good question. It's a feline, so it could be lions as well. Right. But yeah, nobody knows why specific cats.
Not just domesticated cats.
No, any kind of cat. And in fact, it turns out many of these parasites, their definitive or their sexual cycle is they sort of zeroed into a particular animal. So malaria, Plasmodium, which causes malaria, is a relative of Toxoplasma. It falls in the same Apicomplexa, that's the phylum Apicomplexa. They also have a sexual cycle, and that takes place in the mosquito. And not only in any mosquito, only the female mosquito, which is the mosquito that actually feeds on blood, basically. And so, and for some reason, the mosquito is the transmitting organism, and so it makes sense that it would happen in a mosquito. That's where the evolution happened. So in cats, I've no doubt that the reason why it happens in cats is not because of the interplay between cats and humans. It probably had to do with feline and whatever organism at some point had the predecessor of Toxoplasma in it, and that's where that sort of developmental pathway evolved. There's another parasite called Neospora canidae, which is a relative of Toxoplasma as well, and it has a very similar phenotype, but its sexual cycle takes place in a dog, not in the cat. So, and you name it, you find any organism, there's one that the sexual cycle takes place there. Yeah.
I do. I don't want to take you too far afield, but
No problem.
I had heard that, so the other part of the life cycle, right, involves mice.
Mm-hmm.
And that I had heard that the parasite makes the mice not as afraid of cats.
Yeah.
And then I heard that some people think that like it makes you like cats, or it makes you like a crazy cat lady.
Yeah. So that's a really interesting. So that's actually really interesting, and there are a lot of studies, not a lot, but there are a few studies going on where they're trying to understand the connection between toxoplasma infection in the brain and whether that has any psychological effects. And there's this guy in his last name is Felger, I think. He's from the Czech Republic, who's done a lot of this work. And he received, I don't know, you've heard of the Nobel Prize, right? Have you heard of the Ig Nobel Prize? So there's a prize given by Harvard called the Ig Nobel Prize, and it's given to people who've designed experiments which are scientifically sound, but they're kind of ridiculous. Like, for example, somebody who won a few years ago for, I can't remember, it was psychology or something else, but it was, what is the effect of, zapping an artist with an electric rod while they're painting a polar bear or something, some crazy stuff like this. They followed scientific method, and then they came up with some conclusion. Like, why did they do this experiment, right? So this guy won the Ig Nobel Prize just because a lot of the work that he produces is not really conclusive that there's a direct link between toxoplasma infection and a psychological effect. So, for example, if we go down to your high school and you got the authorization to give all the high school students a questionnaire, right? And you ask them many questions about their IQ, maybe about their aptitude and different things, and then you took blood from them and you tested if they have antibodies against toxoplasma, right? If they have antibodies against toxoplasma or anything else, it means that at some point your body was exposed to that organism and your body generated antibodies to defend against the organism. It doesn't mean you're actively infected. It doesn't mean that toxoplasma got to your brain. It doesn't mean anything except that at some point in your life you were exposed to toxoplasma and now you're able to elicit immune response against toxoplasma. Okay, so you have your questionnaire and you have your antibody response. And now you go in and you say, oh, okay, it looks like everybody who scored low on the IQ, there's a higher percentage of people who scored low on IQ also have a higher percentage of having been exposed to toxoplasma, right? Are these, linked? Are they causative? No, they're correlative. Yes, there's a correlation between having antibodies against toxoplasma and having a low IQ, for example. But you don't know that this causes that, right? You could argue the other way around, that you have a low IQ, so you play with, cat food. It depends how you play. So anyway, so there were many studies that do that. So taking a step back, though, there are some really good studies taking place in mice and rodents, where they've specifically examined whether the rat, for example, has a cyst in its brain. And then they determined whether they are less or more afraid of, let's say, the smell of cats. Right? And so there's some evidence that, yes, it turns out they're less afraid of cat urine smell, for example. The problem with those experiments is that they don't really say that they're more likely to be eaten by a cat, right? So the real experiment is to actually, but it's inhumane, right, to do that experiment and then see if a cat is more likely to eat a rat that's infected with toxoplasma versus not. There are some really good examples in nature, however, where parasites have commandeered, an insect or an animal to go and climb up on the highest leaf on a plant so that it gets eaten by the bird, so it gets transmitted. So there are examples of those co-adaptation things. And in this case, there may be something, but it's not clear yet. So, yeah, but I mean, certainly, there's again, not causative, but the correlation between the crazy cat lady and having hundreds of cats in your house and being a bit slightly awkward or slightly off, maybe being exposed to lots of episodes of invasion of toxoplasma in your brain, it could have some effects, but we have no evidence that it's really happening. So. Okay, so any other questions about sort of toxoplasma biology? It's one important thing. It's haploid. Right, so it has a single copy of every chromosome. It has 14 chromosomes. We're diploid, right? We have 23 pairs. So that's sort of a difference. The only place where toxoplasma is diploid is during the sexual cycle in the cats, when it's exchanging DNA. Right? But besides that, it's a haploid genome. it makes it, haplogenomes are easier sort of to when you're studying biology and trying to sort of understand genomes and stuff like that, it's fairly easy because you don't have to worry about the second chromosome, basically. You're always looking at one chromosome at a time. Okay? Other questions or any other questions?
You said if one is a vegetarian, then they still will probably have this toxoplasma inside of them. What about, you can buy spinach today and it says it's been triple washed. Would it be your recommendation to still wash it again?
Oh, so I do. I personally wash myself just because, well, I was once talking to somebody in a conference who visited some of these plants where they triple wash vegetables. And she said, oh my God, if you see the vats where they triple wash these things in, they look so disgusting. I have no doubt that they're clean. I just, out of habit, I always rinse because I'm more, not because of the potential parasites and organisms, more because they're probably using some chemicals on there, so I'd like to sort of give them an extra rinse to get up whatever chemicals they use maybe to rinse those. But that's just a habit of mine. I'm sure they're clean. And in terms of why vegetarians, people would ask, well, why are vegetarians? Why could vegetarians, if you're vegetarian all your life and all you've eaten is plants, why could you be infected with toxoplasma if toxoplasma is transmitted, by eating meat, right? That's one mode of transmission. Well, cats, it's transmitted through their feces, and then cats, they go everywhere, right? And so even on plants, and so and actually in the dirt, you can find oocysts of toxoplasma in dirt, in soil, and so that's the way it'll be transmitted via the soil, basically, on vegetables that are not properly washed, basically, or properly handled. So, yeah.
If someone wanted to get I believe it's called an antibody titer, or would that show whether or not you have antibodies for toxoplasma?
Yeah, you could do that. You have to order a special test, and I wouldn't recommend anybody doing it just out of curiosity because it's just going to make you worry, and you can't do anything about it really, and it's in most likelihood it'll never have any effect on you whatsoever. So my recommendation is out of practice. I won't just go around doing titers just to check on that. If you're concerned and you're worried that you're getting sick or something, but I would talk to your doctor and they can order that for you. But I don't think it's covered by any insurance, so you'd be paying extra for it.
Are you starting to collect cats?
No.
Okay, so now getting back to the database, and let's make sure I'm doing this on time. Yeah. So this is one of the databases, so toxodb.org, from just by looking, there are lots of options, lots of things you can click on. So then don't have to worry about getting bogged down by it. But basically, the three things I would concern yourself with is that there are three sort of main columns here. You can search for genes, identify genes by any of these criteria. You can search for other data types. And I probably won't talk too much about these today unless you're really curious, and I'm happy to go into these. And then finally, there are links to a number of tools that you may want to access. Depending on what you're doing and how advanced your class is, you may decide you want to go further. So we do have a module for you, which is a genome browsing module, which is using a resource within this database. But you shouldn't feel stuck with only doing that. If you have a class that's really inquisitive and they want to go beyond that, there's a lot more in this database that you can explore and would be quite exciting. And I want to demonstrate one of these sort of in silico experiments. We're going to do an in silico, or an experiment in the computer, basically, and just to show you how those work. And then we'll switch to the actual genome browsing module, and we'll probably have you guys work on that, and sort of we'll circulate. And we're going to be doing an experiment on genes. Okay, so we're going to ask questions about genes. And so genes, as you know, they can have, so genes are transcribed into RNA, and then the RNA is translated into protein, right? And each of these steps has different features, right? A gene could have multiple exons, or it could be a single exon. The transcript could have untranslated UTRs, upstream and downstream untranslated regions, right? And so again, don't get bogged down by the terminology. We'll get into that when we look at the genome browsing. But the bottom line is that every gene or a gene can have characteristics. And those characteristics, if those have been annotated on the genome, you can look for them. You can ask questions about them. So for example, if a protein is secreted from a cell, right? Lots of proteins like insulin is secreted from cells, right? If it's secreted from a cell, it has something called a signal peptide, which lets you, tells the protein, it's like a zip code telling the protein, you go out of the cell, all right? And it turns out you can actually search for those. There are algorithms that actually identify those signal peptides, and we can search for them. So what we're going to do is we're going to ask, show me all genes in ToxoDB. So ToxoDB has multiple genomes in it. It has toxoplasma, but it has many other related genomes in there. And so I'm going to go in and I'm going to search for all genes based on cellular location. And I'm going to ask if they have a signal peptide. And you get to a search page where you can define your different characteristics. We're not going to worry about this, but there are multiple organisms in here. There's Eimeria, which infects chicken. Hamandia, I think, infects cats. The Eurospora chicken, dogs. Sarcocystis is in horses. Toxoplasma, various mammals, including humans and cats. And I'm just going to leave the defaults, right? I'm just going to click on Get Answer just to illustrate what this gets you back. And so when you run this search, what happens is you get this little box here that tells me I found about 30,000 genes in ToxoDB, so from all of these organisms, that have a potential signal peptide, a predicted signal peptide. So these are 30,000 genes that are potentially secreted from these parasites, right? And since these parasites invade our cells, secreted proteins are really important, right? Because they are going to go out there, they're going to interact with the proteins in our cells, and they're going to modify them. They're going to, they're the things that are at the interface in this, in this battle between the parasite and the human cells, right? So they must be, if they're secreting them, they must be doing something. You would assume they're doing something important for the ability to infect and survive in the host. 30,000 is a lot of genes, right? If you're going to do an experiment in the lab and you want to work on 30,000 genes, depending on what kind of experiment you're doing, it's a lot of genes and very hard to start sort of testing a hypothesis on these genes. So the idea is you'd like to sort of narrow down these 30 genes into something a bit more manageable that you can work on in the lab. And so the cool thing about this method here is that it allows you to add steps to this in silico experiment. We call it a search strategy. And it allows you to sort of build a logical, stepwise searches that are linked together. And so, for example, I can add a step, and I can say, well, under cellular location, I noticed when I looked at this, there's predicted signal peptide, there's also transmembrane domain. So a protein could have parts of the protein, right? The protein is made out of amino acids, and the sequence of amino acids, each amino acid itself has characteristics. They could be polar, they could be hydrophobic, they could be positively charged, negatively charged. And depending on whether you have a certain series of them in a row, they would give the protein a particular characteristic, a three-dimensional structure, an ability to do something, right? So, our membranes, our cell membranes are made up of lipids, right? And for a protein to cross the membrane barrier and anchor in the membrane, that part of the protein has to have some specific characteristics that allow it to do so. You can't just take any protein and try and shove it through a membrane. Physically, it just won't work. So that protein will have to have certain characteristics. And it turns out, if it has something called a transmembrane domain, it is something that allows you to trans and go across membranes, right? And so, and you can search for those. So what I want to do is I want to say, well, out of those 30,000 genes that have a signal peptide that they're growing out, how many of them also have a transmembrane domain? Okay, I want to narrow down my search. So I'm going to select transmembrane domain. Again, I'm going to keep this at the default. I don't care. I just want it to have at least one and as many as as it could have. And now I have to decide how am I going to combine my searches. All right. My first search was for signal peptides, right here in the yellow. And my second search is for transmembrane domain. And now I need to join my two results. Okay, so imagine you have two columns in an Excel file, and I'm saying, okay, show me anything in this column that's also in this column, right? That's basically what I'm doing by this sort of fancy graphical interface. So in this case, I'm going to use an intersect, right? And so these graphical icons allow you and guide you through the whole process. And now when I do this, you'll notice here that there are two steps in my experiment. My first one was for signal peptide containing proteins. My second one is for transmembrane domain containing proteins. And my intersection is what has both. So I have almost 10,000 genes, right, down from 30,000, 10,000 genes that have both of these characteristics in one, okay? And now you can imagine building on this and making this strategy as long as you want and narrowing down your genes. And so this is sort of a nice graphical way, especially these days, you know, many of our students are very sophisticated. They can program very quickly and do lots of things on the computer. And so this could be potentially a nice way to graphically illustrate to them, you know, how you can take characteristics of genes and intersect results and come up with some interesting findings. Go ahead.
How long did it take to make that bus to design that? Or is it still being?
It's actively, we continuously actively develop it. It's a team, so we have a team of software engineers and user interface people that work together. And we've been in operation since around 2002. And it's evolved over the years. I mean, if you saw the initial, what this looked like, you know, many years ago, it didn't look like anything like this. But yeah, and you know, many of these resources, and this is actually a nice thing. You know, when you pay your taxes and you know, the trash collection comes every Monday or every Tuesday or whatever day it is, and they come and actually pick up your trash. That's great. That's an amazing thing when you see that. You say, wow, this is amazing that it works. You know, you may argue, oh, but they don't, you know, they don't really clean up all the trash. They throw half the trash on the ground. But ultimately, they're coming and they're picking up the trash every week, you know, on the dot. And you know, and if it snows, they come the next day and they tell you if they're coming the week after, right? Yes, we complain because yes, it would be nice if they pick it up when I want them to pick it up, but of course we can't have everything. But it's a nice illustration of your tax money actually working to something, right? That you see an effect of. And this is a nice illustration, I think, again, from the scientific aspect where your tax money is being effective, because ultimately the reason why this resource exists and the way it exists is because we communicate with our scientists very heavily. So we actively communicate with our scientists and we hear from them what they want. And they tell us, oh, we want this, we want that. Can you develop this, can you develop that? And we put it together ultimately so they can actually do their experiments. And it's actually really nice to then go into the scientific literature and find out that people are using this resource and citing in the paper and saying, oh yeah, you know, in order to do this experiment, we had to use this resource to ask these questions, and then we did the experiments and we had.
That would be another question. Who's using that? A lot of people.
Yeah, so we have, it's a huge, so actually for all of our resources I can show you, so we have, I won't go through the details right now, but if you're curious, you know, late at night and you can't fall asleep, there's, on the bottom here, there's user stats, and you can actually go in and see, let's see, where is it? There it is, website statistics. So you could go here and there's a bunch of user statistics. You can go and see where, you know, who's using the database and the resource stuff. So we're heavily used, so our resources are cited almost 1,000 times annually in papers, which is quite significant as far as online resources go. So it's actually really significant. And so it's good. So, and most of the scientists are people who work on parasites, obviously, because we are a resource for people who work on parasites. And they're all over the world. So a big bulk is here in the US. Again, it depends on the funding, how much funding there is for research. The US is a country that is endowed with a lot of funding for research, which is great. And as a result, we have more people using these databases, right? If you go to places like sub-Saharan Africa, where malaria is endemic, but the resources are very, very poor, and they're worried about actually, you know, how they're going to control the mosquitoes, you know, you're not going to get a lot of usage of the database. At some point, we hope that will happen, but right now, it's not there yet.
With respect to the mosquitoes that carry malaria, is it a viable solution to that problem to have everybody sleeping in nets?
So that's one solution. So the solution to malaria is there's no single method that could eradicate malaria. Many things have been tried. So I think there are multiple things that are a problem for malaria. One is regional stability, right? And so if countries don't have stable governments and a stable economy, then you can't sort of have a public health system that can enforce rules and provide adequate treatment and bed nets and so forth. I mean, you would be surprised to know that malaria was endemic in the Philadelphia region up until the 1930s and 40s. There was malaria here in Philadelphia. George Washington had malaria, right? And Washington, you know, one of the suggestions to combat malaria in Washington, D.C., was to build a massive net around the entire city, and that was actually what's proposed in Congress in the,
Donald Trump.
well, yeah, exactly, his ancestor. But yeah, basically, it's been actually a problem for a long time, but the only reason why we were able to eradicate it here is because obviously the economy is a stable government and a good economy, and then you're able to enforce and implement public health initiatives that are sustainable. And that's a big problem in many countries that are endemic for malaria, is that they don't have a stable economy and a stable government to actually enforce or implement any kind of public health initiatives, even the bed nets, treating prophylactically with drugs, making sure there are no standing water. There are lots of things that you can do. Right now, the newest idea is to introduce genetically modified mosquitoes that are resistant to malaria and have them replace the population of mosquitoes. There are all these possibilities. That's why the Gates Foundation invests a lot of money in endemic countries, and specifically they look for initiatives that are wholesome, that actually not only accommodate things like a bed net, but also, for example, there were stories of villages being given bed nets, but then nobody's using them because the bed nets are being used as fishing nets, because they need to eat, right? And they make great fishing nets. And so it's clear that in order for them to use the bed nets, they also have to have a way to provide for their families and eat, right? Because ultimately you're going to make decisions based on what's more important to you at the moment. Getting sick with malaria later on, this seems like far away from you. Okay, all right. So, okay, so this was just to illustrate that you can run these types of searches and build strategies. And if you're ever curious, there are a number of public strategies that you can take a look at. Let me see if I can open one of these that could be interesting. So I'll see if anyone, but just to illustrate how long one of these strategies can be, right? So this is a group that ran a bunch of searches. They intersected them, and they came up with one gene that is potentially of interest to them. And then they may want to go back to the lab and work on it. And so what this sort of illustrates, how you can combine these searches together and ask these questions. And the questions vary. So the type of data that's available in these databases is not only the genomes and the annotation of these genomes. So each genome, when it's sequenced, then at some point it gets annotated, where you can go into the genome and look for genes and say, oh, right here, from this location to that location, there's a gene. And then you can say, oh, and this gene, based on its characteristics and based on its similarity to other genes, it's insulin, or it's this, or it's that, right? And so, you can do that computationally. You can annotate a genome, or you can do it manually, which is painstaking, takes many years. And so that's one layer of data that's on a genome. But then you can actually do an experiment like an RNA sequence experiment, which we will explore today in the genome browsing module, which allows you to look at the expression levels of these genes. Right? Remember, a gene is transcribed into RNA, and usually the transcription of the RNA is regulated developmentally or based on the conditions you exist in. So your cells in your body are not expressing every single gene at every single moment in time. That'll be a big waste of energy, right? Because it takes energy to express any of these genes and it takes resources, right? And so your cell has to decide, at any moment in time, do I need to make this gene? Do I need to make the transcript of this gene in the protein? Do I need it right now? And so, or not, because if I don't need it, I'm not gonna make it. I'll save my energy and do other stuff, right? And so, and that's the same thing that these parasites are doing. They're this type of decision making, not real decision making, but those types of processes are taking place in the parasites as well, and they have to decide or know which genes need to be turned on at what time and so forth. And you can assay for that. You can run an experiment called an RNA sequence. Did you guys do anything about RNA sequence today already? Okay. So you can run an experiment and analyze RNA sequence data and look at how much RNA sequence you have for a particular gene. Okay. Okay. So I think it might be a good time to move to the module. So are you guys ready for that? All right. So. All right, so this is, Okay, so the module we're going to run, and I'll encourage you, so everybody has a laptop here, and I think what I'd like to do is have you guys walk through it. And if you're comfortable working in groups, that actually works very nicely. If you sit down next to somebody else and you guys work together, and I'll sort of walk around and talk to you individually as well, and I can also demonstrate the whole thing maybe in the end or throughout. But I think it would be nice to get started on it. And the idea behind this module is you're going to go visit. So I'm assuming everybody has the link to this module, yes? And we're going to do, so the gbrowse, actually let me do gbrowse 1 with you. Let me describe the Genome browser first, and this way it's, you're familiar with that. And then we'll do the gbrowse module 2 as a hands-on exercise. So I'm just going to quickly take you here back to ToxicDB and show you what the Genome browser looks like. All right, so the Genome browser, so there, imagine Google Maps basically, right? So when you're looking at Google Maps, what you're doing is you're browsing the world, right? You're browsing the world and you're looking at addresses, locations, streets, traffic, and you may look at distances between one place and another place. You may look for restaurants, but these are all things that are decorated on a map, basically. And a genome browser is essentially a Google Maps for genomes, right? So instead of looking at a map, something we're familiar with, you know, you go down the street, you know what streets are and so forth, we're looking at genomes, right? In this case, in the genome browser, and this case is the genome browser of Toxoplasma, we're looking at chromosome. I just randomly went to a location in Toxoplasma. We're looking at chromosome 6, okay? And this chromosome it starts from 0 and goes all the way to almost 4 megabases in size. So 4 million nucleotides, right? That's the length of this chromosome, chromosome 6. And we are only looking at this little part here of chromosome 6, okay? And the zoomed out part of chromosome 6 is shown right here. And each of these little graphics represents a gene in that section of chromosome 6. Okay, so if we zoomed out, so I can click and drag across chromosome 6. Well, let's make it maybe a little less. The more data you try to load, the longer it takes. But basically, if you zoom out to a bigger portion of a chromosome, you're going to see many more genes displayed in that portion of the chromosome, right? Typically. So you can see, you can barely see anything. It's just genes everywhere, right? So let's go back and zoom into another location. All right, so now I'm looking at another small location of chromosome 6, which is located here. And that's, you can see here, it starts at 880,000 all the way to 960,000, right? So close to 100K nucleotide region, 100,000 nucleotide region of the chromosome, and these are all the genes that are displayed in that region. You can mouse over any of these and it gives you more information about them, right? So there's a lot of information here, you know, don't get bogged down about the details, but it's essentially, this is the information about this particular address, right? So if this were your house, this would be the information about your house. It'll tell you something about how many cars are parked in front of it, what are the colors of the windows, does this house have a name, you know, does it have a specific roof, blah, 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 you know, you'll find all of this. So this is kind of information about this gene. And, you know, as you go down, you'll see some of them have more involved names that mean something to scientists, and other ones, like the one I was looking at just a second ago, just says hypothetical protein, which means we know nothing about it, right? Was there your question?
That was my question.
Okay, yeah. So it just means that algorithmically, if you look in that region of the genome, it has the characteristics of a gene, right? It has what looks like a start site for a protein. It looks like it has a stop site for, you know, the termination codon. It looks like it has characteristics of exons and splice junctions, all these things that you can look for in a genome. And the algorithm that looked for these genes found one here, but then when it tried to compare it to anything else in the databases, say, is there anything out there that looks like me? It said, no, there's nothing out there that looks like me, right? So all I can tell you is hypothetical, that's it. But it is there, but it's hypothetical, right? And so, so often these are very exciting for scientists because they go in and they say, oh wow, this is a gene. If you ran one of these in silico experiments, and in the end you had a hypothetical gene, that could be very discouraging and say, oh, I thought I'm going to find something that something is known about it. Or as a scientist, if you're trying to discover something new, it could be very exciting because you say, wow, this gene met all the criteria I looked for and nothing is known about it. So I'm going to go and research it and find something about it because I think it's very important, right? So you notice some genes are in blue, some are in red. This is simply showing you the orientation of the gene, right? Is it going from the left to the right or from the right to the left? And this is arbitrary. It's based on what is the 0 of your chromosome and what is the last digit of your chromosome. And when they sequenced it and assembled it, they had to decide this part is 0 and that part is whatever, 4 million. There's no real reason why a chromosome should start from this side or that side. At least as far as we know, there isn't anything like that. Okay, so this is purely the numbering system is for humans to actually try and understand organize the chromosome in a linear fashion, basically. Okay, you can zoom in closer to any of these. All right, and you'll get a bit more details. You'll notice that, for example, I'm looking at 2 genes here, both going in the same orientation from left to right. And you'll notice that they're blocks, and then they're lines. Blocks, line, block, line. So each block represents an exon, and each line represents an intron. So this is going to be spliced out of this gene when the messenger RNA is spliced, okay, into something that's going to be transcribed into or translated into a protein. Okay, does that make sense so far? Okay. So in the genome browser, so there are many things you can do in the genome browser, but one important thing you can do is you can select additional tracks. So we're looking at a track here that's the chromosome, another track here that's the zoomed in region of the chromosome, and then we have a track here that has the genes in this region of the chromosome. But you can add additional information on here, and the information is plentiful. So like I said, we integrate a lot of data into our resources so scientists can actually ask questions about them and look at them and view them. And so right now this is all collapsed, but any one of these can be expanded, and you can open these, and each one has data that you can select and display. So it really depends on what kind of question you're asking. In the module, we're looking at the question asks you to go to the transcript expression section and look at RNA sequence data. There's a lot of RNA sequence data. I'm just going to pick one. Let's just pick one randomly. I won't pick the one we're going to be doing because it's described in the experiment. But once you select it, it immediately starts loading it in the browser. If I go back to the browser tab, now you'll notice here there's another track. Here are my genes, but now there are these 2 tracks, new tracks that loaded. And this is RNA sequence data. Did you guys look at a graph, similar graphs like this today? It's basically, all of this is you have a y-axis and an x-axis, and this simply tells you how much RNA was found from this region, from these samples, from this experiment, okay? And so the higher it is, the more RNA that was found. So the way, again, just as a reminder, the way when you do an RNA sequence experiment, you experimentally, you take the sample out of your test tube, you extract the RNA, right? You break it up into small pieces, and then you take that RNA, those little small pieces of RNA, and you sequence them, okay? The method of sequencing is really irrelevant. There's high throughput sequencing, there's low throughput, there's next generation, second generation, third generation, whatever you want. But they're all methods that allow you to determine what are the nucleotides, one after the other in your pieces of DNA that you're sampling, okay? Now, once you have those nucleotide information, it's a jumble of little pieces of sequence, okay, in a file. Now you have to somehow figure out, well, where do those belong? Where do they go? Well, luckily, for many of these organisms, we have a genome sequence, right? So now you have your RNA sequence, which is little pieces, and you have your genome sequence, and you're trying to say, okay, how do these little pieces relate to my genome sequence? Right, so what you need, obviously, you can do it manually. You can go into each one of these, look at the sequence and say, okay, there's an A, a T, a G, a C. Okay, now I'm going to go to my genome, my millions of nucleotides, and I'm going to look for that same sequence, right? Of course, it'll take you years upon years upon years to do that, because some of those experiments, those RNA sequence experiments, you're getting on the order of 100 million reads, right, from an experiment, right? So those are 100 million little pieces that now you need to match up to the genome somehow. So luckily, we have computers with big processors which can take those and align them and look for their matched pair, okay, where they belong in the genome. And I don't know if there are trucks here. So think about it. If you have your genome, right? The genome will have a potentially annotated gene on here, right? So let's say we have a 2-exon gene, okay? So this is one exon, this is a second exon, all right? If this is transcribed, so this is the genome here. So if this is, let me turn it around. There we go. Is that better? So when this is transcribed, these exons are going to be made into RNA and they're going to be spliced together. So what it's going to look like in your RNA, it's going to essentially look like this, right? Here's exon 1, here's exon 2. This is 1, this is 2, right? So this is your RNA. And when you do RNA sequencing, ultimately you took this, you broke it up into small fragments. There's, you do different methods to process the samples. And then ultimately the readout of the next generation sequencing or this high throughput sequencing is all these little fragments, right? That came from this gene and every other gene that was expressed in that particular time point in that particular experiment, right? So now you're going to take these, and the only way for you to know where they belong is you're going to have to try and map them back to the genome, right? So you're going to be able to take these and you're going to say, oh, this one belongs here, this one belongs here, this one belongs here, and so forth. So you get to do this. Some of them actually may be broken up here and here because they were on the junction, right, between where these 2 exons came together, right? So that's an important thing you can think about when you talk about alternative splicing. This can play a role or can be important there. But ultimately, you have essentially a pileup of reads, okay, on each of these exons. And each one of these peaks here is one of these pileups, basically. It's all of these reads that map. And so this is a log scale, a log 2 scale. But if you change it to a linear scale, it'll be thousands of reads that are mapping to any one of these regions. And if you look at this a bit more closely, and in fact there's a ruler here which you can click on and drag it down here, and you can see that anywhere there's an exon, it looks like there's a peak of RNA sequence. Do you guys see that? So it goes from here to here. So for example, from here to here there's a peak, and it coincides with this exon. There's a peak here, and it coincides with this exon. There's a peak here, it coincides with this exon, right? Which makes sense. If this gene is being transcribed into RNA, and you sampled this RNA, right? Then all the little fragments, when you map them back, they will not map. Of course, when you map them back, you're not going to get mapping. You may get some mapping here randomly, or maybe there's some gene that is undiscovered, because you can discover new genes. But most of the mapping will be to the exons, right? Because those are the things that are going to be ultimately make it into the transcript that you assayed in your experiment. Is that clear? Any questions about this?